All right, so for this last video on chapter 19, we're going to go over the white blood cells, and then we're going to go over platelets, and then we're going to take a look at what happens when you have damage to blood vessels. How does your body prevent you from bleeding to death? So let's take a look at the white blood cells. Another name for the white blood cells are leukocytes. Leukocytes. Okay, and so there are 5 different types of leukocytes that we want to look at. There's neutrophils, eosinophils, basophils, monocytes, and lymphocytes. And what we'll notice is that white blood cells have nuclei, and they also have some other organelles, but they lack hemoglobin. So they look very different than the red blood cells. They're very different than the red blood cells. In each one of these blood smears here that you're seeing, you can see a red blood cell, and then you can see a white blood cell mixed in with the red blood cells. So a typical microliter of blood contains 6,000 to 9,000 white blood cells per microliter. All right, so there's that many white blood cells. Most of the white blood cells at any given moment in your body are more in the connective tissues or organs of the lymphatic system rather than in the blood. So we're not going to see a whole lot of these leukocytes floating around in the blood. So let's go through each one of these and talk about some of the differences between them. We're going to start with the neutrophils. So first of all, we have to kind of know which one of these blood cells, white blood cells, is the most common, which is the least common, and the percentages of each of them. So a really good mnemonic that you could use would be never let monkeys eat bananas. All right, so this is going to tell us which is the most common and which is the least common. So by using the first letter of each word, N for neutrophil, L for lymphocytes, M for monocytes, E for eosinophils, B for basophils, we can remember which is the most common, which one we have more of, neutrophils, which one we have the least of, basophils. So let's go ahead and look at these. We'll start with the neutrophils. Neutrophils make up 50 to 70% of all circulating white blood cells. They are the first to arrive at the sign of infection, right? So they're the first ones there. They're very, very fast, and they're very active, and as soon as they get there, they are phagocytes. They're phagocytic, so they are going to just start engulfing anything that looks abnormal. They're going to engulf bacteria that have been marked with antibodies. So they're just going to gobble these things up while they're waiting for other cells to come along. Then the eosinophils, the next one that we see, and so when we're looking at these, we can see these big old nuclei, right? They're really odd-shaped nuclei, okay? When we saw that here in this neutrophil too, really big nuclei, right? All right, so now we're on to the eosinophils. Again, they're like very not common, and they make up about 2 to 4% of the circulating white blood cells. They are going to defend the body against parasites and flukes, like parasites like flukes, and other parasitic worms. Right? So if you were to go to South America and eat some vegetables that hadn't been properly cleaned, for example, and you got a parasitic infection, then these eosinophils would start to increase in number. I mean, they try to actually engulf these parasites, but a lot of times the parasites are too big to be engulfed. So then the eosinophils will shoot out little chemicals that will hopefully burst holes in the flukes and the worms. So eosinophils are phagocytic, but sometimes these things that they're trying to eat are too big. The other thing that eosinophils do is they increase whenever you have an allergic reaction. So if a patient were to have an allergic reaction to a particular drug, for example, and you drew their blood, you would see an increase in the eosinophil count. Next, we have the basophils. The basophils make up less than 1%. So, right, they are the least common of all the white blood cells. They discharge, if we look at them, we can see this big old nucleus, right? And it's right there. It's huge, right? And then we look at the cytoplasm, and in the cytoplasm, there is a lot of granules in there, right? There's a whole bunch of granules in there. And those little granules in there, they secrete two chemicals. One is called histamine, and the other one is called heparin. Histamine and heparin. And so histamine and heparin are going to help with inflammation. They actually cause inflammation. Histamine dilates blood vessels. So when your blood vessels dilate, they bring more blood to the area, right? So, and then they make the blood vessels more permeable. So now more fluid, more blood is coming to the area. The blood vessels are more permeable. So fluid leaks out of the blood vessels and into the interstitial space, or the interstitial tissue, right? So that's inflammation. It's causing an inflammation in the interstitial space. And then heparin, what heparin does is it prevents blood clotting. So this heparin then prevents blood clots from forming. All right, then the next white blood cell that we have is called a monocyte. A monocyte accounts for 2 to 8% of circulating white blood cells. So monocytes here are like right in the middle. And so monocytes are big. They're big, and when they're floating around here in the blood vessel, we call them monocytes. But these monocytes like to squeeze out of blood vessels, and when they do that, that's called diapedesis, right? So they squeeze out of the blood vessel. Diapedesis actually means squeezing. And now they're out into the tissues. And once they're out into the tissues, we stop calling them monocytes and we then start calling them macrophages. So these are phagocytic cells, these macrophages, and they're going to try to engulf things that don't look quite right. So they're very, very aggressive phagocytes. And they are going to eat up to about 100 cells that don't look right. So they'll eat 100 bacteria or 100 infected cells. So they eat up these cells and then they finally die. But they've done their job. They've tried to really clean up the area. So they're very aggressive. And then we finally have these lymphocytes. Lymphocytes are the second most common. Lymphocytes make up 20 to 30% of all white blood cell populations. Most of these are found in connective tissues, and they're also found in organs of the lymphatic system. We're going to talk about the lymphatic system in a couple of weeks. Actually, next week, we're going to talk about the lymphatic system. It's part of your immune system. There's three different types of lymphocytes. There's the B cells, there's the T cells, and then there's the natural killer cells. And we'll go into that more into depth when we get to the immune system or lymphatic system. So we do want to talk about the production of all of these cells, your red blood cells, your white blood cells, your platelets, and where they're produced. The white blood cells are going to be produced from these cells that we call hemocytoblasts. So hemocytoblasts are these omnipotent cells that divide to produce two other stem cells. The hemocytoblast cells, which are stem cells, can produce myeloid stem cells, and they can also produce lymphoid stem cells. So the lymphoid stem cells, they can only produce lymphocytes. That's the only thing that they can produce. So if the hemocytoblast differentiates, it divides, and then it differentiates into a lymphoid stem cell, the only thing that it will be able to become at that point is a lymphocyte. However, if that hemocytoblast differentiates into a myeloid stem cell, now it can become a number of things. First of all, it can become an erythrocyte, which is a red blood cell. It goes through several different stages, which we talked about in a previous video. The myeloid stem cell could also maybe become a platelet. So first of all, the myeloid stem cell gets really big and has all these extensions and becomes a megakaryocyte. And then pieces of the megakaryocyte break off, and those little pieces then are called platelets. So a myeloid stem cell could also become a platelet. And then finally, if a myeloid stem cell doesn't become an erythrocyte and it doesn't become a megakaryocyte, then it could become one of the other types of white blood cells. So it has to differentiate into these other types, and it can do that. It will become a basophil or an eosinophil or a neutrophil or a monocyte. What it will not become is the lymphocyte. So what I want to talk about now then are platelets. So if we look over here and we see these platelets, they're not full cells. They're not cells at all. As a matter of fact, they're only fragments of a cell. They are fragments of the megakaryocyte. So let's talk a little bit about platelets. So again, platelets are just fragments of the megakaryocyte. Okay, and what they do, their function is to initiate blood clotting. They initiate blood clotting. They'll circulate around, so they circulate for about just a little bit over a week, for 9 to 12 days, almost 2 weeks, and then they die in the spleen. Now I'm going to show you in the next slide when we start talking about blood clotting what platelets actually do. They form what are called platelet plugs. And they help you to stop bleeding so that you don't bleed to death. So the platelet plug is just a temporary patch. And then that temporary patch has to be packed down with fibrin in order to become an actual blood clot. And then after the blood clot forms, then it also has to be retracted so that we can still get blood flowing to where it's supposed to flow. Now, the production of platelets is called thrombocytopoiesis. So poiesis always means production of. Cyto means cell, and thrombo stands for a thrombocyte, which is another name for a platelet. So thrombocytopoiesis, right? So that thrombocyte is a platelet. So thrombocytopoiesis is the name we give for the production of platelets. Now let's go on and talk about the process of stopping blood loss, which is called hemostasis. So hemostasis is the process of stopping bleeding. Hemo means blood, stasis means stop. So this is the process that stops bleeding and it prevents the loss of blood when the blood vessels have been damaged. There's some major steps that occur during hemostasis. The first step is called the vascular phase. So in the vascular phase, let's just say you have a blood vessel that's been cut. Okay, so it gets cut right there. We'll just do it in black, like a knife went right through there and cut it. So two things are going to happen during this vascular phase. We're going to get vascular spasm and we're going to get sticky ends. So this phase is going to last about 30 minutes. So what happens, let me just show you what's going to happen here. The vascular spasm means that right where that blood vessel has been cut, there's these smooth muscles in the walls of the blood vessel. They're going to contract and they're going to make that hole much smaller than what it was originally. So that's what we call vascular spasm. We go from this wide down to this wide. So that's the vascular spasm. The next thing that we get then is a stickiness. Now on the ends over here, these cells have been broken, and so this little cytoplasm leaks out and it just makes those ends really sticky. Okay, so that's what we get in the vascular phase. Now, if this were just a small capillary, this phase alone might close off the passageway. So these endothelial cells here might just kind of stick together and then it just closes off the pathway and the bleeding stops and then that's all that needs to be done. But if it's not, if it's in a little bit bigger vessel than a capillary, capillaries are extremely small, then we get to the second phase, which is called the platelet phase. Now remember what those platelets are. Those platelets are little fragments of that megakaryocyte, right? So those platelets are normally just floating around in the blood like they're supposed to. And let's just say that we got a cut right through there. And we ended up getting a little bit of a vascular spasm and sticky ends. Okay, so we get vascular spasm right like that. And then the ends get really sticky, like I told you they would, right? And along come those platelets. So those platelets are traveling through the blood, and then the platelets are going to start to stick. So they start to stick right there like that. Okay, they're sticking. And so what we end up getting then is what we call a platelet plug, where the platelets have plugged up the area and now blood can no longer leak out. Because otherwise, blood would be leaking out this way, but that's not what happens, right? We don't get that leaking out anymore because of this platelet phase. So this usually starts pretty quickly after an injury, like 15 seconds after an injury. And the platelet plug, again, might close off that break in that vessel wall if the cut's not very severe. But if the cut is severe, then this process of hemostasis might enter into the last phase, which is called the coagulation phase. Coagulation. Okay, so in this coagulation phase, we have the platelet plug here. Right, so we've got all these platelets in there, plugging stuff up. And it's looking like it's a good temporary fix, but it's not permanent. And so this happens pretty quick too. It takes about 30 seconds after a cut for this to start. But in this process, a plasma protein. So in the plasma, remember in the blood here you have plasma and you have cells, blood cells, right? And then you also have these plasma proteins. Well, one of these plasma proteins that we have is called fibrinogen. Fibrinogen. And when this occurs where we get a cut in the blood vessel wall, it's going to trigger this process where fibrinogen will convert to fibrin. Right? So there's some other steps involved. We're going to go over those different pathways in advanced, but for right now, it's just we need to know that fibrinogen converts to fibrin. Now, fibrin looks like these little fibers here. So now we've got these little fibers in the blood, and then these little fibers get to where this platelet plug is, and they start to intertwine their way in between all of those platelets, those little fragments of cells, and pretty soon what we end up with is what we call a blood clot. Okay? So this blood clot occurs. Now, there's a positive feedback here because as the blood clot occurs, it will produce more chemicals that will convert fibrinogen to fibrin, and that will then cause the blood clot to release even more chemicals, and then fibrin makes more of a clot, and it just keeps going on and on and on until finally the blood clot is big enough to shut off the entire blood loss. So that is what we call positive feedback. It's positive feedback because the process continually gets more exaggerated instead of stopping. We get more fibrinogen being converted to fibrin because this blood clot is forming. So that's the coagulation phase. Then after the blood has stopped leaking out of the blood vessel, what we'll find is that the endothelial cells of the blood vessel will kind of grow around that blood clot and connect to the other side, right? So we just kind of grow around the blood clot, connect to the other side, and now that blood vessel's intact again. But now we have to get rid of that blood clot. This clot retraction is sort of the shrinking of a blood clot. So it's clot retraction. And it's where the blood clot starts to shrink away, and it takes place over a number of days. And there are multiple coagulation factors from the platelets that are trapped in the fibrin mesh of the clot that will be released and will help to dissolve that clot. All right, then that is the end of this last video for chapter 19, which is on blood. We'll see you next time.